Welcome back, and as promised, I'm going to pick up where we left off with our osmosis portion of our lecture. So we are in passive transport of molecules across the cell membrane. Remember that passive transport does not require any energy for this to happen. And now we're specifically talking about how water gets transported across the membrane, and that process is called osmosis. And the protein, since water is hydrophilic and very polar, it can't cross that hydrophobic core. No polar molecules can cross the hydrophobic core freely. You just can't wiggle your way through. You need a protein in order to do that, and that process is called facilitated diffusion. And the specific kind of protein that only allows water to go in and out is called an aquaporin. So let's see how this actually works in cells that we're talking about. So we're going to compare the different tonicities. Remember, the tonicity is the ability of a solution, and a solution is when you have water mixed with something. The ability of a solution to cause a cell to gain or lose water. So remember, when we're talking about tonicity, we're referencing the solution that the cell is in. Not what the cell is, but what the solution is inside or outside the cell. So there's three types of tonicity. You have hypotonic, isotonic, hypertonic. And I'm going to show you an example for an animal cell and for a plant cell. So animal cells will be on top, plant cells will be on bottom. Let's talk about hypotonic. So if I'm a cell floating around in a hypotonic solution, that means, in comparison to me, the solution outside of me has a lower solute concentration. There's less stuff outside of my cell than there is inside of my cell. Now remember the direction that water wants to flow. It's a little bit counterintuitive, but if you think about it, water just wants to spread things out. That's what water likes to do. It doesn't like things to get concentrated, it likes to spread it out. So if there's lower solute outside of me and higher on the inside, water is actually going to flow into the cell. Now this could be dangerous for an animal cell because, take for instance a red blood cell that's just floating around and doing its job. If a red blood cell is just hanging out and doing its job and it's in a hypotonic solution, that means that there's low solute on the outside, and that which means it has to be higher in comparison on the inside. So since there's too much on the inside, water is going to flow into this cell. Our cells, animal cells, all animal cells, were not made to withstand too much water. Think of a water balloon. You put too much water in it, it's going to burst. And that's exactly what happens to an animal cell. It actually kind of bursts open and dies, unfortunately. We call that lysed. So whenever you see lysed, we talked about lysis, like hydrolysis with our reactions when you break apart something. Anytime you hear about lysing happening in the medical world, that means things are literally blowing up and they're not supposed to do that. But, different story if you're a plant. If you're a plant cell, you have this cell wall surrounding your plant cell. And on the inside is your cell membrane. Now, if you remember, the cell wall of a plant cell is made of a bunch of cellulose, all these fibers that are just making it nice and rigid. When a plant is getting water put into it, that's a good place for a plant to be, because what that does is that plumps up the membrane against the cell wall, and that causes plants to stand and do what they need to do. If you're like me, hopefully not, I'm not the best with my plants. My husband takes care of all the plants in the house, which I'm very thankful for. I forget to water them. So what happens when I forget to water them? Bad things happen. We're going to talk about that in a moment. But when I do water them, if I remember to water them, they automatically get plumped up and good things happen, because when I forget to water them, the solute concentration is really low on the outside and it gets super high on the inside, because I've dehydrated that poor plant cell because I'm not giving it water. So when I do give it water, water flows right in and plumps up the plant. And that's a happy place for a plant cell to be. We call a plant cell that's in this scenario, we call it turgid. It's nice and tough. That's a good place for a plant cell to be. Now let's move on to isotonic. Now remember, iso means equal. So that means you have equal solute concentration on the outside of the cell compared to the inside of the cell. So if I'm a lovely little red blood cell again, that means it's equal on the outside, equal on the inside. I got water going in, and I have just as much water going out. That is a happy place for our animal cells to be. Our animal cells don't like to be in anything besides isotonic. When we get out of our isotonic range, bad things can happen. That's why if you've ever had to get an IV at the hospital or known someone who've had an IV at the hospital, that they don't put straight up water into you. That would be awful, because then you would be in a hypotonic solution. Does that make sense? They always mix it with a little bit of salt, because inside our cells we have salt as well. So that keeps our tonicity in an isotonic state, and that's healthy for our cells. So that's a great place for an animal cell to be in. When you think of a plant cell, not so much. Plant cells, you got your cell wall, and things are just starting to wither down. This is where my plants hang out most of the time, unfortunately. But you can see how the membrane is actually starting to shrink in compared to the wall. So this is the cell wall, and this would be the membrane. So in an isotonic solution, you have just as much water going in and just as much water going out. You have this equal exchange of water. That's not allowing the plant cell to remain plump and do its job. This is when your plants start to become limp. It's actually the technical term for the whole entire thing. It's not just they're starting to fall over, which is sad news for the plant. But it's still recoverable. You just got to water that thing. All right, lastly, the hypertonic solution. So if I'm on a hypertonic solution, it's the exact opposite of my hypotonic solution. That means I have a high solute concentration in the cell compared to the inside of the cell. And inside the cell, it's really, so here's my cell, it's really low, right? So what the cell wants to do, it sees this high solute concentration on the outside. Water naturally just wants to go to where there's a lot of stuff. So water is going to escape the cell. And what eventually happens is this cell is going to just shrivel up and die. We actually call it shrivel. And that's bad for your cell. So either extreme for the animal cell is really, really bad. You want the happy middle for your animal cells. Hypertonic solution for a plant cell is also just as bad. This is when water is leaving the plant cell. You can imagine that just doesn't make sense for a plant cell. It doesn't like that. And so even the plant cell wall starts to buckle in on itself, but then even the membrane just starts to collapse and does all this stuff, and the whole thing just starts to shrink in. You can see how here, when you're in a hypotonic solution, the membrane is pushing against the cell wall. As we start losing water, because it's not coming in, and now it's starting to leave as well, the membrane is starting to shrink in. Now that tons of water are beginning to leave the cell because we're in a hypertonic solution, it's high on the outside, water is escaping this plant cell, and that's causing this poor little plant cell to just shrink up, and we call that wilting. So, wilt. So if you ever hear anyone say, oh, that plant is wilted, that's bad. Most plant cells, depending on their environment, if they're indoor, outdoor, they might be able to recover to where they used to be. Plants are very hardy, especially if you train them to be that way. Animal cells, not so much. It depends. You might be able to get from shriveled back to isotonic, but you can never recover from being lysed. It's not good. So just to remember, the happy places for a animal cell is in isotonic, and the happy place for a plant cell is in hypotonic. Okay, that ends the passive transport portion of our notes. And what I want to do is start introducing the active transport part, because it'll take a couple, or it'll be one more lecture after this as well. So get ready, let's do a little active transport. So that was the end of passive transport. Here we go. So big number 2 in your notes. Number 1 was passive, so number 2 is active transport. Now you could probably already assume what the definition for active transport is, and you're probably right. If passive transport is the transport of molecules across the membrane without the use of energy, active transport is transport across the membrane requiring the use of energy. And here's why. Whereas passive transport, things are just diffusing along the gradients. Even water is doing what it does naturally. It's going from a region of low to high solute concentration, which is weird, but that's what water does, and it makes sense because water wants to spread things out. But if I have a whole bunch of oxygen on the outside of my cell, oxygen is going to diffuse on to the inside where there's not as much. So that's diffusion. Active transport, this is when you need something to go from a region of low concentration to high concentration. You're going against the flow, you're going against the gradient. That kind of push across the membrane requires energy, and that's why it's called active transport. So number 2, active transport. This is transport across the membrane that requires energy because the transport of whatever we're doing, the transport is against the gradient. And here's what I mean by against the gradient. You're going from a region of high solute, forgive me, oops, going back, from a region of low solute to high solute. You're going in a way that doesn't seem natural for this molecule to go, but it has to be done. So before we get into the different types, or one great example of active transport, we need to talk about the energy, how we get our energy. So the one thing we need to know is a term called energy coupling. So energy coupling, it's the process that cells manage their energy expenditure. So in order for me to make something move, so what's actually truly happening in the cell membrane, there's a protein that's going to grab something in a low concentration and move it across the membrane to a region of high concentration. That movement of the protein is pretty neat. The proteins actually move. They do what's called a conformational change. Those conformational changes, those changes in their shape, if you will, need energy. And where they get this energy, you just can't make up energy anywhere you go. There's a molecule that when it undergoes a reaction, it actually releases energy. The energy released from that molecule goes onto that protein to allow it to move. And so energy coupling is basically the coupling of a energy producing reaction to enable an energy requiring reaction. So the coupling of an energy producing reaction and if you want to go technical with this, if you ever go to take an SAT test or an AP Bio test or anything like that, energy producing, that word is exothermic or exergonic. Either one, it depends if you're going bio or chemistry, just depends. So if you're making an exothermic reaction, you're producing energy. So it's a coupling of an energy producing reaction. Oops. With a energy requiring. So it needs energy in order to happen. Energy requiring reaction. And that is called endothermic. Or endergonic. So essentially, I can't move, I can't do something that, because I need energy. So it's like almost as if in order for me to move from this end of the board to that end of the board, someone has to come and shove me, because I'm not going to be able to do it myself. That's energy coupling. And our cells work like that all of the time. So where we get our energy is a lovely molecule called ATP, adenosine triphosphate. You gotta love ATP because we're going to come across it a lot more later on. So let's talk about ATP for a second, and then we'll wrap up this portion of our lecture. So little b under this. So little a was energy coupling. And now we could talk about where we get all this energy. Little b is ATP, or adenosine triphosphate. So what adenosine triphosphate is, here's how it works. And the structure of it may seem a little familiar. Here's what, I'm not going to draw the structure of adenosine, I'm just going to put it like that because it's a nitrogenous base. Is this coming back to you a little bit? I hope so. And we're not done yet, but that should hopefully remind you of a nucleotide. And it should, because they look almost identical. But ATP has triphosphates. There's 3 phosphate groups. So you have 2, 3. That is ATP, a very simplified view of ATP. These phosphates, if you remember correctly, they are negatively charged. And if you have 3 negative phosphate groups next to each other, do you think they really like being next to each other? These similar charges are repelling each other like crazy. And so what happens with ATP, so ATP naturally, there's enzymes involved in here that allow this process to happen. What ATP does, it cleaves, boom, that last phosphate off to become ADP, adenosine diphosphate. And now that phosphate group that just got released is pure energy. So if you could imagine essentially like 3 big cats in a box, in a way. I don't think they want to be in a very small box. They don't want to be together. You let one of those cats out, that cat is going to run and do something crazy. That's exactly what that phosphate does. That phosphate is now, it's pure energy. This phosphate is going to binds to proteins to allow work. So as soon as, if I'm a protein and that phosphate group binds onto me, I can now move, okay? Without that phosphate group binding onto me, I can't do the conformational change that needs to be done. I need that shove, I need that push, I need the energy from phosphate to do that. And that phosphate energy comes from adenosine triphosphate. This whole process, this is hydrolysis. So when we say we hydrolyze ATP, we do a hydrolysis reaction. Some water comes in and goes cut right here. It cuts off that phosphate, turns that ATP into ADP, releases that phosphate that's full of energy. That phosphate now finds a protein to go help it do its work, essentially. And when a phosphate binds to something, we call whatever that thing is, it is now phosphorylated. So this whole process is called phosphorylation. Okay? So I want you to appreciate how awesome ATP is, because in a couple units we're going to talk about how we make ATP. Without ATP, our proteins can't move. And without our proteins not moving, we're not doing anything. So ATP is awesome and it has to happen. There's a lovely reverse reaction to convert ADP back into ATP, so we don't ever run out of this stuff, but that's coming later on. So I hope that makes sense. This is where the energy is going to come from to do our active transport. Now come back, I'm going to show you one example of what active transport looks like. It's the classic textbook example of active transport, and it'll make how the importance of a phosphorylated protein allows a protein to move and change shape to allow something to go against its gradient in the cell. So thank you, come back, I'll see you soon.